then we move online now to Dr. Johnston. He is working at Health Canada, working on vaccines and especially on the regulatory or improving the regulatory process for approving those. So please, Dr. Johnston, go ahead.
Thank you very much. This is not an EV talk, it's a LNP talk, and it's discussing our work on our prototype Lyme disease vaccines. And just our standard disclaimer, information and views expressed at this presentation are those of the presenter and should not be taken to represent Health Canada's views or policies. And generally, information provided in this presentation is taken from publicly available sources. So a bit of a background on the project. We noted an increase in development of LNP vaccine systems in 2017, 2018. In early 2019, we initiated a research program looking at LNP vaccines for RSV and Lyme disease. And then obviously in 2020, that included expansion into the pandemic. And most recently, we've started looking at LNP influenza vaccines. And the goal of our project was to develop tools to improve our understanding of these systems. And we thought the best tool that we could develop was functional prototype or model lipid nanoparticle vaccines. And we were doing this in support of anticipatory regulatory research, assisting in the development of assays for the assessment of these products, supporting mechanistic studies of vaccine function, expanding our roles of the specific functions or components of these vaccines, and then to provide ourselves with some understanding of microfluidic manufacturing and long-term storage of these products. So the first disease we looked at was Lyme disease, and it's a bacterial arthropod vector disease. In North America, it's spread by deer ticks. Currently, there's no approved vaccine. One was withdrawn for a number of reasons in 2002. And generally, early detection and treatment with antibiotics is considered effective. However, missed or delayed treatment can result in chronic neurological, cardiac, and rheumatological manifestations. And what's concerning us is the northward spread of the vector that's been attributed to climate change. So this is a map of the exposure risk to deer ticks carrying the bacteria. This is produced by the Public Health Agency of Canada. And as you can see in 2005, the darker red indicates a higher chance of exposure. And by 2015, and this is borne out with actual clinical cases, we can see the northward spread. And what's concerning for Canadians, although Canada is a very large country, a little over half the population lives in this region here. So we have a large number of Canadians potentially exposed to Lyme disease. So we feel that a development of a Lyme disease vaccine is imminent or will progress very rapidly. So this is the data presented today was published earlier this year. Because of the short time, if someone's interested in more details, I direct them towards this publication. So we took the outer surface protein C, which is a protein expressed on the bacteria during the early stages of mammalian infection, and persists for about 3 weeks after initial infection. So we felt it was an ideal vaccine target. Took a type A OSPC, cloned it into a PVAX1 vector, encapsulated that in a standard LNP. Encapsulation efficiency was about 99%, and particle sizes were about 75 nanometers in diameter. Our study consisted of a prime and a boost, followed by bleeds 3 weeks after each inoculation, a challenge with a homologous bacteria that expressed the type A outer surface protein C, and a necropsy at the end of the study. We initially observed significant increases in anti-OSPC IgG, which favored a Th1 response. We also noted that the serum from our vaccinated mice was bactericidal. So when we cultured these spirochetes in tissue culture, we noted the serum induced about 80% reduction in bacterial growth. After the challenge, we assessed the presence of the bacterial flagellin B, which is a motor protein for the spirochete, in a number of tissues, and found our vaccinated animals had no detectable levels of the bacterial motor protein. We also did microscopy looking for bacteria at either the site of injection, which was in the leg, intramuscular in the leg, or distally in the ear. And again, we found no bacteria in either of those sites in our treated animals. Finally, we examined the heart and lymph nodes. So what we observed was no detectable levels of carditis in our vaccinated animals, and no enlarged lymph nodes in those as well, whereas the unvaccinated animals had mild to moderate carditis and elevated levels of white blood cells in their lymph nodes. So we demonstrated that vaccination of these mice with our DNA lipid nanoparticle vaccine targeting the outer surface protein C type A provides effective protection against homologous needle challenge and resulting pathology. So we observed obviously significant levels of anti-OPSI antibodies, predominantly by a Th1 bias. And the major limitation of this study was that the outer surface protein C is highly heterogeneic. So there's over 20 variants of that protein, and 4 of those variants have been associated with invasive infection of the bacteria. And we feel that a human Lyme disease vaccine should obviously confer protection against heterologous challenge. So our future studies will include a very large animal study looking at potential cross protection, assessment of multivariant formulations or multivalent formulations of this prototype system, and moving to an mRNA platform. Our initial studies were done with DNA. When we started the project, we had issues obtaining significant quantities of mRNA material, so we went ahead with DNA. But now we can obtain large quantities of mRNA coding for these outer surface proteins. So I'd like to thank my lab and then my collaborators, and specifically Casey, who's did most of the formulation work for this study, and then within the vaccine lab, 2 excellent grad students, Annabelle and Alicia, who did the animal work. So with that, I'd like to thank you for your attention, and I'm willing to answer any questions you may have. Thank you.